And what I mean by that is, if you have a little chunk of flesh for some reason that gets ripped off your body, the fibroblast cells are the ones that go through mitosis quickly to fill in that missing chunk of your skin. So normally they're not dividing, but if you get some flesh ripped off of you, then they get back into the cell cycle to fill in that missing bit. Liver cells are like that also. If something causes you to lose part of your liver, the remaining liver cells can get back into the cell cycle and replace that part.
You said that we never produce more muscle cells; they just get bigger.
Yeah, that's correct. When you're, if I think of little infants or babies, they have the same number of muscle cells we do, but those muscle cells just haven't grown to their full size yet. But they don't get new muscle cells.
Is there like any research been done to try to get a cell to jump out of GO phase?
Yes, I'm not sure exactly where it's being researched, but if they could convince nerve cells to regrow, they might be able to make people walk again who are in wheelchairs. And I'm sure a lot of laboratories are trying to work on that, repair brain damage and severe injuries to the spinal cord like that.
So when your cells grow larger, say when you're working out, they get larger and your muscle cells get larger. When you stop working out, do they contract? And
yes, I believe they do. That the muscle cells are extremely filled with proteins. They're extremely protein-rich cells. And so I think what the body says, if the person's not using those muscle cells for exercise, it starts using the protein inside them as a protein reserve. So if the body needs amino acids somewhere else, it will start breaking down the protein inside those muscle cells and will start getting smaller again. Oh, okay. I just suddenly realized why I couldn't find the phrase. I think this is this acetate handout I'm projecting here is not the right one. This is from a previous version of this lecture that I didn't update. So your handout might say something slightly different from this. Go with what your handout says, not with what I'm projecting up here. Oh yeah, these are totally the wrong handouts. Let me glance real quick and see if I can find. Yours says this?
Yeah.
Oh. Does anybody have something that says something different? Because I thought I posted my updated ones. Maybe I didn't. Yikes. Well, darn it. Well, I'll go with this for now, but if your handout says the exact same thing as this, it's not supposed to. So I'll double check that I posted the updated version of this. Most of the definitions are the same, but they're not exactly the same. Just out of curiosity, to show of hands, does anybody have something that's different than this? Okay, so I think most of you got the updated handout. Alrighty. Where was I? Oh yeah, so many of the cells in your body, in fact, most cells in your body, are in this not dividing category. Here we go. Here, this is supposed to be your skin. Here's the outer surface of your skin right here, and here's that deeper layer I was talking about right there. You don't have to know the name of it, but it's called the stratum basale. And anyway, there is the cells are constantly dividing to replace the ones up here at the top that are constantly flaking off. So those guys are constantly within the cell cycle. Here's a nerve cell, not dividing, and it can't divide. It's permanently stuck in GO. And here are some of those fibroblast cells that are in GO, but they can re-enter the cell cycle when it's appropriate to. And there's a scar from when those fibroblasts divide and fill in the missing bit of flesh. All right. Now, yeah, this does not really match up with what I want to say right here, but when a cell like a fibroblast that is parked in GO needs to re-enter the cell cycle, like when you cut yourself, how does it figure out that it's now supposed to start dividing? Well, these cells that are in GO, but that can re-enter the cell cycle, have a receptor on their surface, a receptor protein on their surface, that can bind things called growth factors. And growth factors are signal molecules that cause cells to re-enter the cell cycle from GO. Here we go. That green thing is supposed to be the growth factor, so some other part of the body makes the growth factor. It travels to the cells that are in GO, binds to their receptor. They then are pre-programmed to move past the G1 checkpoint and start dividing. So in other words, they re-enter the cell cycle. Like that. And that leads to the activation of the S-phase enzymes, like the ones that duplicate the chromosomes, and the ligase, and the primase, and all the other ones that carry out S-phase. And there's actually several different, there we go, you see S-phase in operation. There's actually several different growth factors. There's one for fibroblasts, and there's one for, I guess, liver cells also. I don't know exactly how many different growth factors there are, but they're very specialized. Each particular cell in the body has its own particular growth factor to get it to move past its own G1 checkpoint. All right, what about that checkpoint right there at the end of G2? That checkpoint at the end of G2 is there for making sure that the DNA is ready to go through mitosis, because notice right here the cell has not actually entered mitosis yet. The nuclear division part is not started yet, because that will begin in prophase. And so this checkpoint basically stops the cell and verifies that the DNA is ready to go through mitosis, starting with prophase. And what do I mean by is the DNA ready? Well, the major thing is, are the chromosomes duplicated? Because remember that you need duplicated chromosomes to go through mitosis, because the chromosomes are going to eventually get split in half. And so that's one thing that's checked for at the G2 checkpoint. Another thing that the cell checks the DNA for at the G2 checkpoint is the DNA damaged? Because if something has damaged the DNA, the cell wants to repair that damaged DNA before it allows the cell to progress into prophase. Now, what can damage your DNA? Well, just as one example, sunlight can. In sunlight, there's a type of radiation called ultraviolet radiation, UV light, that when it shines on your skin can give you a sunburn. And that's actually caused by the UV light damaging your DNA. It mutates some of the bases. It can even break parts of the backbone. So if you've got a bad sunburn like that, your cells will then halt at the G2 checkpoint until the damage is repaired, and then it can proceed past the G2 checkpoint. So let's say that one is okay. So we now move into prophase and then into metaphase. The metaphase checkpoint is there simply to make sure that the spindle fibers have attached to the chromosomes correctly. Remember, in metaphase, the chromosomes are lined up in the middle, and each chromosome has to have spindle fibers attaching it to the top pole of the cell and to the bottom pole of the cell. There we go. And so it verifies that each chromosome is attached to the top pole and the bottom pole, because that's going to be necessary also for the two sister chromatids to pull apart from each other. And there we go. Now, once a cell has been given the go-ahead to move past the G1 checkpoint, in other words, once it gets a signal to move past the G1 checkpoint, it's only allowed to do one round of cell division. In other words, it will re-stop itself the next time it hits the G1 checkpoint, unless the growth factor is still present. In other words, it automatically stops here and goes back into GO unless the growth factor is still present. And that's good, because that allows the body to control the mitosis in a very controlled way. In other words, the cells will only go through mitosis to the extent the body needs them to, and the body controls that by only providing the growth factors for the proper number of rounds of duplication that the body needs. Sound all right? You buy it? Yeah.
There's a growth factor for one.
Various locations. Like I think the brain makes some. Injured cells will release some special kinds of growth factors that cause the fibroblasts to grow in that area.
And those work as what kinds of single transduction?
Well, that's going to be something I want to probably get to on Wednesday. So I'm glad you asked that question, but it's a little complicated when we dig into it on Wednesday. The pancreas, which is the same organ that makes insulin for regulating your blood glucose, makes some growth factors also. What else? White blood cells can make growth factors if you're fighting an infection, and those growth factors stimulate other white blood cells to grow. So there's a bunch of different growth factors, and they come from different locations.
So, cancer divided is that checkpoint isn't there?
Yes, that's something else I want to mostly defer to Wednesday, but in a nutshell, cancer is where the checkpoint systems break down, and so the cells get stuck dividing even though they're not supposed to be. Okay. So what I want to begin to do now is look at that same topic of getting past the G1 checkpoint and into S phase, but dissect it at a little more deeper level as far as the molecules that are involved in it. There we go. All right, so here comes this growth factor again. It's going to bind to the receptor, and that's going to cause the cell to re-enter the cell cycle, which means move past the G1 checkpoint and get into S phase. Let's see that more close up. So there in blue is the cell, and you see the growth factor receptor sticking up out there. Oh, yeah, again, this is the wrong handout. Well, can I use this one? Yeah, maybe I can use part of it. So hopefully yours says something different than this. Down here I put a little diagram of where we are in the cell cycle just so you can keep track of it. So notice we're stuck there at the cell is stuck at the G1 checkpoint. Where I wrote here the S phase enzymes, this protein right here is going to represent the enzymes that carry out S phase, like DNA polymerase 3 and primase and ligase and all the other ones that duplicate the chromosomes. These are the enzymes that actually carry out S phase. And I drew them in black to show that they are not activated, because if you're in G1, you're not in S phase, and so those S phase enzymes are just not activated yet. And over here is the receptor for the growth factor. What are these guys right here? Well, think back, those are signal transduction proteins. Remember, whenever you have a receptor, there's going to be signal transduction proteins which carry the signal from that receptor down deeper into the cell to activate the enzymes that actually carry out the response you're looking for. And it's kind of a review question. Does anybody remember the name of those enzymes from the lecture on signal transduction? The ones that carry out the process are called the whats? Cellular response enzymes, yes. That was the term we used when we talked about signal transduction. The cellular response enzymes are the ones that actually carry out the reaction that the cell is supposed to have to the signal molecule. And the ones in between the receptor and the cellular response enzymes are called the relay proteins. All right, so these are relay proteins that are involved in getting the cell to move into S phase. All right, so here comes the growth factor. Here comes the growth factor. Here comes the growth factor. There it is. It plugs into the receptor. The first relay protein in many of these signal transduction pathways that get the cell to move into S phase is a protein called RAS, R-A-S. At one point I knew exactly where that name came from, but now it actually escapes my mind. But just call it the RAS protein. So it is the first relay protein. And remember, each relay protein then activates the next relay protein in the signal transduction pathway until you finally get to the cellular response enzymes, in this case the S-phase enzymes. So that's what we're going to see here. RAS is activated directly by the receptor, and then it's going to activate the next relay protein like that, and then that will activate the next relay protein, which activates the next relay protein, all the way down the pathway until you finally get to the S-phase enzymes. Now when we say activating, almost always we mean kinasing. And remember that works like this, that a protein that's activated, that's a kinase, is one that attaches phosphates to amino acids. So a kinase will grab an ATP as a source of phosphates and attach one of those phosphates to the next protein, and that's what activates the next protein, which is usually a kinase itself. And so now that protein, now that it's activated, will typically go off and activate the next protein in the pathway. Now I want to go slightly off the, how am I doing? Yeah. Darn it. Okay, we're running out of time. I'll pick it up again pick up this again on Wednesday. All right, so see you in the laboratory. Don't forget that you'll have a weekly quiz on the 2 labs we did before vacation, which I think were photosynthesis labs, if I remember correctly.